All right, so you've got a big neuro exam on the horizon? Perfect. You are in exactly the right place. We're going to do a super high-yield review of all your sensory systems. We're talking about how you actually perceive the world, how photons and pressure waves turn into the action potentials your brain can actually make sense of. So let's just dive right in. It really all boils down to this one big question. How does your brain build your reality? I mean, think about it. It's taking all this raw data from out there in the world, and it's building this incredibly rich, seamless experience that you live in every single moment. It feels like magic, right? But it's not. It's just a series of really elegant biological processes, and that's exactly what we're going to unpack.
Okay, so here's our game plan. We're going to hit sight, then hearing, then the chemical senses, that's smell and taste, and then we'll wrap it all up with the sensory toad that kind of ties it all together. First up, let's talk about sight. All right, let's start with vision. This is all about how your eyes manage to capture light, which, is really just photon energy, and start that amazing process of turning it into a neural signal, something your brain can actually see as an image. And the whole show starts right in the back of your eye, in the retina. So the retina is built in these really distinct layers, and for your exam, you are absolutely going to want to know these 5 key cell types. It's a little counterintuitive, but light has to travel all the way to the very back to hit the photoreceptors. Then the signal starts moving forward through the bipolar cells to the ganglion cells. And it's the long tails, the axons of these ganglion cells, that bundle together to become the optic nerve. So what about the other two, the horizontal and amacrine cells? Well, you can think of them as the editors. They handle the side-to-side or lateral processing that helps sharpen up the edges and boost the contrast of everything you see.
Okay, here we go. This is a classic exam question. Rods versus cones. It's all about how these two photoreceptors split up the work. Think of rods as your night vision specialists. They are incredibly sensitive to light, but the trade-off is they only see in shades of gray, black and white. Cones, on the other hand, are your daytime high-definition specialists. They need a lot more light to get going, but they're the ones that give you all that sharp, detailed color vision. And that's exactly why they're packed super tightly, right in the fovea, the dead center of your vision.
So how does a single photon of light actually create a signal? It's all through this process called the phototransduction cascade. Now here's the key thing to remember, and it's a little weird, a little counterintuitive. In total darkness, your photoreceptors are actually on. They're depolarized and constantly releasing glutamate. So when a photon comes along and hits the cell, it kicks off this whole chain reaction that, get this, closes an ion channel. That stops the positive ions from flowing in, which hyperpolarizes the cell. It basically turns it off. And that reduction in glutamate is the signal. It's the brain's way of knowing, hey, light is here.
Okay, think about it logically for a second. You have millions of these ganglion cell axons, and they all have to leave the eyeball somehow to get to the brain, right? Well, that exit point, where they all bundle together, is called the optic disc. And because it's basically just a big cable of neural wires, there's literally no room for any photoreceptors. And, well, no photoreceptors means no light detection. It's a blind spot. It has to be there.
But hey, don't just take my word for it. Let's actually find your blind spot right now. Go ahead and close your left eye, and with your right eye, stare directly at the cross on the screen. Don't look away from the cross. Now, just slowly move your head closer to the screen. Or maybe a little further away. Keep going. And at a certain point, boom, that dot on the right is just going to disappear. That's it, you found it. That's the image landing squarely on your optic disc. And what's amazing is your brain is so good at just filling in the blank space you don't even notice it's there.
All right, vision check. Let's switch gears and move on to hearing. So we're going from light energy to mechanical energy now. Hearing is this totally incredible process where your ears take physical pressure waves, literally just vibrations in the air, and translate them into neural signals your brain can understand as sound. Now, before that vibration even gets to the really sensitive parts of your inner ear, it gets a massive boost. Those 3 tiny bones in your middle ear, the malleus, incus, and staples, they act like a perfect little lever system. They take the tiny vibrations hitting your eardrum and amplify them by a factor of 200. That's what creates enough force to actually get the fluid moving inside your cochlea.
And deep inside that cochlea, that's where you find the organ of Corti. This is really where the magic happens. As that fluid sloshes around, it makes these tiny membranes inside the organ move. And that movement physically bends these little hairs, the stereocilia, on top of the hair cells. And that tiny physical bending, that's it. That's the trigger for the entire auditory signal. Pretty amazing.
Okay, hold on a second. If you've been hitting the books hard for your neuro exam, this next part might make you do a double take. We're taught over and over that potassium leaving a cell, efflux, causes hyperpolarization, right? Well, in the ear, it's the complete opposite. Potassium causes depolarization. So, what on earth is going on?
And here's the solution to the puzzle. It's all about the unique environment. The hair cells are sitting in this special fluid called endolymph, and it is packed with potassium. It's so high in potassium that it completely flips the normal concentration gradient. So, when those little channels get pulled open by the bending hairs, potassium doesn't flow out, it rushes into the cell. And that rush of positive charge is what depolarizes the cell, makes it release glutamate, and sends that signal to the brain. This is a super important detail to lock down for your exam.
Okay, let's move on to the chemical senses, and we'll start with smell, or olfaction. What is just so cool and so unique about smell is its pathway to the brain. It's like it has a VIP pass. It is the most direct route of any of your senses.
Seriously, take a mental snapshot of this slide. This is a guaranteed exam question. Every single other sense we're talking about, sight, hearing, taste, touch, has to send its information through the thalamus first. The thalamus is like the brain's main sorting center or switchboard. But smell? Smell gets to bypass all of that. It has a direct, private line straight to the olfactory cortex. And that's probably why certain smell can just instantly trigger such powerful memories and emotions, way faster than anything else.
So how does it work? Well, the mechanism here should look a little familiar. It's another G-protein coupled receptor, or GPCR cascade, very similar to what we saw in vision, just with a few different characters in the story. An odor molecule binds to the receptor, and that activates a unique G protein called GOLF, G-O-L-F, for olfaction. That kicks off a whole chain reaction, producing something called CMP, which then opens up a channel. Positive ions rush in, the neuron depolarizes, and bang, you get an action potential.
All right, and our final sense for this review is taste, or gustation. With taste, we're actually decoding molecules that are physically dissolved in our saliva, right there on our tongue. You could say it's probably the most intimate of all our senses.
This table is a fantastic way to break down the 5 basic tastes. You can really split them into 2 groups based on how they work. First, you've got salty and sour. These are the simple ones. It's super direct. The ions themselves, like sodium for salt or hydrogen ions for sour, just flow right through an open channel. Done. But then you have sweet, bitter, and umami. These are more complex. They rely on those GPCRs we've been talking about, just like we saw with smell and vision.
And here's another one of those high-yield facts you'll want to file away for the exam. Once that taste cell is activated, this signal has to get up to the brain, right? Well, it does that by traveling along 3 different cranial nerves. You've got the facial nerve, number 7, the glossopharyngeal, number 9, and the vagus nerve, number 10. Which nerve it uses just depends on where the taste bud is on your tongue.
Okay, let's pull the camera back now and look at the big picture. We've gone through 4 completely different senses, right? They're all detecting totally different things from the world around us. But they all share one single fundamental goal: convert that outside information into the language the brain speaks.
So for a final quick-fire recap, here are your primary players, the specific receptor cells for each sense. These are the cells doing that critical job of transduction. You need to know these cold. For sight, it's the photoreceptors, your rods and cones. For hearing, it's the hair cells. For smell, it's the olfactory receptor neurons, or ORNs. And for taste, it's the taste cells in your taste buds.
And if there is one single thing you take away from all of this, let it be this. Your brain doesn't actually see light. It doesn't hear sound. It has no idea what a chemical floating in the air is. All of that stuff is just energy and matter out in the world. The only thing your brain understands, the only language it speaks, is the pattern and the rhythm of electrical signals, of action potentials. That is the universal currency of your entire nervous system.
So, I'll just leave you with this one last thought to chew on. We know for a fact our senses can be tricked, right? Optical illusions, auditory illusions, they happen all the time. And we now know that all they're really doing is interpreting signals with a very specific set of biological tools. So, what does that actually tell us about the idea of an objective reality? The one that's supposedly out there, separate from our perception? It's something to think about, isn't it? All right, that's it for our review. Go crush that exam.